All right, everybody, welcome back. We are continuing on to our last section for Module 4, and that is the concept of speciation. So we've gone through adaptive evolution, non-adaptive evolution, and now we're going to talk about, okay, so we know that there are these mechanisms of evolution. How do we actually get to a new species? What makes a species in terms of biology? So your book has this great example, again, looking at the microenvironments of Manhattan. And this study noticed that on all these different street medians, again, they're very isolated. They're almost acting like islands. There are specific and different species of ants that occur on these islands. So ants aren't able to travel between these different medians. So it's hypothesized that these ants developed specifically on these islands and created specific species on, again, these street medium islands. But in terms of biology, what is a species? So essentially, it's a populations of individuals whose members can interbreed and critically produce fertile offspring. And that's really the key, is the ability to produce fertile offspring. So different species cannot mate, and ultimately they're what we call reproductively isolated. So what do we mean by reproductive isolation? Now, I think the first thing that sort of comes to mind automatically is something called ecological isolation. So that Arctic fox and the desert fox are never going to encounter each other. Therefore, they're never going to be able to breed and produce a new species of fox or breed together and create viable offspring. So that's what we're thinking about with ecological isolation. But there are several other types of isolation. For example, temporal isolation. So different species of frog might have different times when they mate. And so again, they are never going to be able to produce viable offspring because their mating is not synchronous. Additionally, you can think of things like behavioral isolation. Again, we see this with Darwin's finches in a video that I'm going to have you watch for your quiz. These types of prairie chickens don't have similar mating displays, and so they're not attracted to one another, and therefore, again, are not going to mate. A couple other ways that we think about reproductive isolation is mechanical isolation. So there are specific flowers that only hummingbirds can pollinate because of their specific long needle-nosed beak, versus bumblebees are never going to be able to pollinate these flowers because of this mechanical isolation. Next is much more on these biological levels. So we're thinking about gametic isolation. So the gametes, and again, we're going to talk more about reproductive biology in later sessions, but basically the sexual organs of these two, the sexual gametes cannot unite between a cat and a dog, so you can't create a zygote, which is ultimately what's going to form an embryo. Then you can have something called hybrid inviability. So basically the gametes are able to unite, but viable offspring can't form. So a sheep and a goat can mate, but that zygote never is going to form, and it does not survive. And lastly, we can have something called hybrid infertility. So where we're able to, the gametes unite, an offspring is able to happen, but this hybrid offspring, in this case between zebras and horses, cannot produce offspring of their own. So they are in fact sterile. So these are the types of reproductive isolation that we think of when we're talking as biologists. So ultimately, the genetic divergence of populations, a lot of what has led to the divergence of species is this idea of reproductive isolation, and that ultimately forms new species. And again, we're going to look at Darwin's finches. You can see that on different islands we have different birds, and they were able to adapt very specifically to those island environments, and those traits that we were adapting, again, influence their ability to reproduce with the birds that their original population may have come from. And so they are then said, because now they have they are gametically isolated, are said to be a new species. So I want to go through just a whole summary of what we've gone through for Module 4. So first off, we saw that a population is identified by a particular collection of alleles in its gene pool. We know that genetic diversity is reflected by the number of alleles, again, the different types of variant genes that are circulating in the gene pool. And then evolution is a change in those allele frequencies over time. And evolution can be adaptive or non-adaptive, in this case, non-random and random. Mutation, genetic drift, and gene flow are all non-adaptive forms of evolution. Founder effect and bottleneck effects are types of genetic drift. Gene flow, however, is the movement of alleles between different populations of the same species. And I'm going to have you watch a specific video on hybrid Hardy-Weinberg equilibrium, just because I thought it did a really fantastic job of explaining this. But Hardy-Weinberg equilibrium describes the frequency of genotypes in a non-living population. Non-evolving population, sorry, not non-living. And lastly, what we talked about today, where speciation can occur when gene pools are separated, gene flow is restricted, and populations then diverge genetically over time. So that's going to be summing up for Module 4, and we will start next week on our new section after we have our test on Monday. So good luck, take a drift, take a drink of water. It's all going to be okay. I promise this test is not going to be as terrible as you all think it might be. We'll just go through it nice and slow. Review your lecture slides, review the quizzes, and it's all going to be great. And we'll see you next time for starting our next section.